Então, né, nós vamos começar a nossa sessão hoje, né, com a presença do professor Dr. João Duarte, que é professor sênior da Universidade de Lund, na Suécia. Ele é líder da equipe de pesquisa e pesquisador principal do grupo de diabetes e função cerebral. Ele é pesquisador principal da ExoDiab, que é excelência em pesquisa sobre diabetes na Suécia, pesquisador sênior associado do WCMM Wallenberg Center for Molecular Medicine, pesquisador principal Multipark, pesquisa multidisciplinar focada na doença de Parkinson. O professor João vai trazer a aula de título A Memória do Diabético. Então, professor João, a palavra é sua, fique à vontade para fazer a sua apresentação. O professor João vai fazer a apresentação dele em inglês e as perguntas no final. Fique à vontade, professor. Obrigada pela sua presença.
Obrigado pela introdução, obrigado pelo convite e é um prazer estar a fazer esta apresentação. Eu vou fazer em inglês simplesmente porque é muito mais fácil para mim, mais fluido, mas no final as questões podem ser em português e podemos discutir em português. Ah, o meu laboratório trabalha principalmente com modelos animais de diabetes, então nós tentamos descobrir mecanismos de como é que a doença impacta o cérebro, como é que a diabetes impacta o cérebro, e é sobre isso que eu vou falar hoje. Mas nós temos algumas experiências que temos feito, ou algum trabalho que temos feito com humanos também, e depois talvez para uma próxima possamos mostrar algumas dessas coisas. So I'll move to English. It seems from the introduction that I do a lot, that I work in a lot of places, but in fact I'm just the head of the Diabetes and Brain Function unit, and this unit it's actually belonging to several different centers here at Lund University, and that's why there's all those titles. But in fact, I'm just associate professor at the university, and I lead this group of very smart students and postdocs that do some work in diabetes. So. So this is just the first slide to tell you that there are many complications of diabetes, as you know, and we are particularly interested in the brain, and this is an interest I had for a long time, since already my PhD started 20 years ago. And in fact, as you see here in this slide, we still don't acknowledge well how diabetes impacts the brain. So we still, or most people still think that the major issue for the brain is the occurrence of stroke and cerebral vascular disease. So that's not what I will talk about today. Of course, those are problems, but the diabetic brain is affected in many other ways, and we'll talk about that today. This is a slide I like to show from a paper 3 years ago, where the researchers looked at the incidence of or let's say deficits in several cognitive domains in the data set from the UK Biobank. And what they did was to look at aging. What are the cognitive domains impacted by aging? And they found these 5 significant domains that are affected. It doesn't matter which they are. That's not the purpose of this slide. But what is important is that if we look at the effect of diabetes, and that's this second graph here, you can see that pretty much there's an effect on the same cognitive domains caused by diabetes itself. So this is corrected for aging. So the effect of diabetes on the same domains is apparent. And in that study, what the authors did was to pull data from many publications, did a meta-analysis comparing diabetes individuals with individuals that are cognitively healthy. And they actually see that there are way many more domains that can be observed impacted by diabetes when you increase the sample size to about tenfold of what is done in a single study. And this is important because it starts telling us that there's no single brain area that could be impacted by diabetes, but it's probably the whole brain that is impacted in a very similar way in the case of diabetes. And I didn't mention it at the beginning, but we work mostly on type 2 diabetes. The effects of type 1 diabetes, they could be involving also developmental issues in the brain, but we nowadays treat the patients with insulin. So and insulin ends up being a growth factor, and therefore could affect development. But that's not so much an issue today because we treat well the patients. But of course, there are other issues with type 1 diabetes, but most of our work is on type 2 diabetes. So how does diabetes impact the brain? Again, vascular dementia is something that happens in uncontrolled diabetes, again, type 1 or type 2, but our work afterwards will be mostly on type 2. And that is because the high blood sugar levels can damage the blood vessels in the brain. And that leads to reduced blood flow and oxygen supply to the neurons, and therefore there's reduced performance in the brain. The other thing that is important is that when diabetes patients are treated with insulin, they might have hypoglycemic episodes, and these hypoglycemic episodes are very dangerous for the brain. So they can, in extreme situations, cause loss of consciousness. So the patients can faint during hypoglycemia. And when there are repeated hypoglycemia episodes, you can also have what is called an awareness of hypoglycemia. So the patients develop a reduction in the sensitivity of the body to sense that there's an hypoglycemia episode coming, so that you don't have the counter-regulatory response as efficient as you should have when you have hypoglycemia. And that becomes an issue also in the long term. But what is most striking and it's being more and more acknowledged, but that we are trying now to understand the mechanism is on how diabetes impacts the neurons themselves. So how the different factors of diabetes, and that let be dysregulation of lipid metabolism, high glucose, the hyperinsulinemia, insulin resistance, how does this impact the neurons, and especially their synapses, because it's the functioning of synapses that makes memories. And that's what we need to understand, and we need to work on that to try to improve memory in the diabetic brain. In the diabetes, individuals with diabetes. I will introduce a cell. I'm not sure what the audience is, but there might be no neuroscientists here. So I'll cover some things that might be a little bit more basic, but I wanted to make sure that that's understood. So I'll talk about the astrocyte. The astrocyte is a glial cell. So the brain has neurons, but has glial cells that support the neurons. And the astrocyte is probably one of my favorite cells in the brain. So the astrocyte has processes that engulf the synapses. As you can see here, this is a presynaptic button. I hope you're seeing my mouse. There's these small vesicles that are ready to release neurotransmitters to the postsynaptic button. And you can also see a mitochondria here in the synapse. And what you have in blue is astrocyte. So the astrocyte has these processes surrounding the synapse. The astrocyte senses what the synapse is doing and can actually modulate the efficiency of the synapse and regulate how the synapses function. That's very important for neuronal function. If you actually impair astrocytic functions in terms of supporting the synapse, you develop impaired plasticity and impaired memory as well in animals. On the other hand, the astrocyte also has these processes that have end feet surrounding the vessels. And that's what you see depicted here. So the astrocyte is in red. The astrocytes are cells with these long processes that touch the blood vessels. Here shown a vessel in green. And you can imagine already that the astrocyte is poised in a way that can sense what the synapse is doing here, and over there can regulate what is taken from the blood flow. So the astrocyte is essential to modulate what is coming from the bloodstream to feed the synapse. So if the synapse needs energy, there will have to be a communication to the blood vessel to tell that, and the astrocyte is involved in that process. One thing that is important is that the more our brains evolved, the larger the astrocytes are. You can see here, this is the same scale in the microscope, and you can see the size of an astrocyte. It's a relatively small cell with some processes. And you see a size of an astrocyte in the human that is much larger, much more processes, much more branching, And this tells us that the astrocyte is able to connect not only to one synapse and one vessel, it can actually connect all over. Plus, the astrocytes are connected with gap junctions, so they form a network that can actually signal and transfer metabolites across different places in the brain. And this is astrocytes stained with GFAP, that is a cytoskeleton protein or associated to the cytoskeleton. But if you actually load the astrocyte with a dye, this is what you see. So this is an astrocyte from the mouse, and this is an astrocyte from the human. So the astrocyte not only has that network of processes, it has a lot of fine branches that are located in synapses and near vessels to regulate a lot of things. It's a very interesting cell. So this is a picture of what I was saying. You have a blood vessel here in red. You have neurons that are connected with synapses in blue, and an astrocyte that is connecting both and operating this communication between the two. And now I want to talk about the glutamate-glutamine cycle, because glutamate is the main excitatory neurotransmission, as it's essential for memory formation. And most of the glutamate is actually not taken back by the synapse that released it, but taken by the astrocytes. So if you zoom in that synapse, this is what you could see. So you have these vesicles with glutamate inside that is ready to be released to the synapse. This glutamate acts on postsynaptic receptors like NMDA receptors, kainate receptors, and so on. And then this glutamate is not recovered by the neuron, but it's taken back by the astrocyte that converts it to glutamine. And this is a key step, because glutamine is not electrophysiologically active. That means that it will not act on receptors in the neuron. So it can be sent to the neuron, where it's then converted to glutamate, repacked, and be ready to signal again. This process costs energy. Taking glutamate comes with sodium that has to be sent out of the cell. That costs energy by the potassium ATPase pump. And converting glutamate to glutamine also costs 1 ATP. So we need 2 ATPs to fuel one molecule of glutamate coming into the astrocyte. And that energy in the brain comes mostly from glucose. So glucose is the main energy substrate used by the brain, and it's used both by the neurons because you need to reestablish the ion gradients after the action potential that released the neurotransmitter. And it's also used by the astrocyte to fuel astrocytic activity that is supporting the synapse. It's noteworthy that the astrocyte is also storing glycogen, or storing glucose in the form of glycogen. And this glycogen needs to be mobilized to support glutamatergic activity. So there are experiments where people blocked the hydrolysis of glycogen by glycogen phosphorylase, and that leads to certain types of memory impairment. So using glycogen in the astrocyte is essential for supporting glutamatergic activity as well. So glutamate, just to summarize here, it's an excitatory neurotransmitter. So the main excitatory neurotransmitter is key for plasticity and memory performance, is the most abundant amino acid in the brain. It's at the crossroad of many metabolic pathways. So almost anything that you can imagine in terms of primary metabolism involves glutamate at some point, and it's made de novo from glucose, and that happens in the astrocyte, because the astrocyte is the only cell. I don't want to diverge much into that, but there's the only cell in the brain that has pyruvate carboxylase that is needed for making de novo molecules of glutamate. Anyway, what happens in diabetes? So we know that when the diabetic brain develops insulin resistance, we have impaired metabolism in the neuron with reduced energy supply to the synapse. We have impaired release of glutamate and uptake by the astrocyte. But on the other hand, we have increased metabolism in the astrocyte. So you generate more energy in the astrocyte. And we also have impaired glycogen metabolism in the astrocyte. Now you question, why do we have this increased energy metabolism in the astrocyte? That is because the astrocytes in the diabetic brain, they become hyperreactive. So they are hyperreactive, and at the same time, they lose the ability of supporting glutamatergic synapses and of supporting memory formation. Next, another cell that is important is microglia. That's another glial cell in the brain. That is the brain immune cell. So it's like the macrophage of the brain. And I'll talk later about this particular cell, because that's also important in the diabetic brain. So as you have seen, let me just go back to this slide. Metabolism is very important in the brain to fuel synaptic activity, especially that activity that involves glutamatergic neurotransmission. And we, in our lab, we are particularly interested in understanding how metabolism is affected in diabetes. So we did, Alba Garcia was a postdoc here in the lab some years ago, did these experiments to try to understand the trajectory of metabolic alterations in the brain while insulin resistance is developing. So how do we induce insulin resistance? You probably know that if you induce obesity in a model with a high-fat diet, this high-fat leads cells to become insulin resistant. And that's also the case in the brain. So what Alba did was to expose animals to a diet that contained high fat and high sucrose. And we did that for a long time, so for about 4 months. And this allowed us to study how brain metabolism is developing during this period. After these 4 months, what Alba did was to split the animals in two groups. One that returns to a normal diet with low fat and low sugar, and another group that persists in this high fat, high sugar diet. You can see here in these graphs the food intake in terms of caloric intake. So when we expose the animals to this high fat, high sugar diet, both male and female mice, they increase caloric intake for 4 months. And when we switch them back to this low calorie diet, they recover the caloric intake. So this low fat, low sugar. And here below you see the weight gain. So females are smaller than males, but they both accumulate fat when they're exposed to high fat diets or high fat, high sugar diets. And you can see the weight gain that starts slightly separating after a couple of weeks. And then after 4 weeks is when you see a massive change in body weight compared to the low fat, low sugar animals. And then what is important is that if we remove this high fat, high sugar, the animals or the phenotype of the animals becomes completely normal. So all the diabetic phenotype that develops by exposing to these hypercaloric diets, it's completely recovered when the diet is returning to normal. And that's what you will see here in this graph. So we have done longitudinal assessment of the animals. The first thing we do is glucose intolerance or glucose tolerance tests. And what you see here is the area under the curve of a glucose tolerance test, where you see that the animals become glucose intolerant already after 1 week of exposure to the diet. And that keeps on aggravating over time. That's much more substantial in males, but it's also seen in females. And if you look at fasting insulin to glucose ratio, you can calculate this OMA-ER, which is a marker of a proxy of insulin resistance. And you can see that insulin resistance only happens after 4 weeks of exposure to the diet. So you first start to have this glucose intolerance, and so only much later you develop insulin resistance. That all, again, becomes severe. And once more, it's happening in males much more than females. And in females, we almost don't see insulin resistance, which is interesting because both males and females develop memory impairment. But what you see in the bars here is that if we normalize the diet, then they completely recover glucose tolerance and insulin sensitivity, males and females. So how do we study metabolism in the brain in a non-invasive way. So we use this magnetic resonance spectroscopy that where we put the animals in an MRI scanner and measure a profile of 18 metabolites from a brain region that we select. And we did it in the hippocampus and cortex because those are areas involved in certain types of memory performance. So we have a spectrum like this, and from here we quantify 18 metabolites. And we can measure these non-invasively. So the animal just goes in and out like a patient in the hospital goes to an MRI machine. Uh, uh, and so in this paper, we actually have a lot of data on this metabolic profile, but I just wanted to show you a principal component analysis of the data. So we try to reduce the number of variables and just look at the profile as a whole. And we can see that early on, there's no real change on the brain metabolic profile in the first 4 weeks. It's only when insulin resistance starts to happen at 4 weeks that we start to see, or after that, that we see that there are brain metabolic alterations happening in the brain. And that's in the hippocampus and also a little bit in the cortex, but to a smaller magnitude. So we did a training of our PCA using the data up to 4 months, and then try to use the model of the PCA to predict the metabolic profile of the animals at the end of the study. And we can actually see that those animals that have been reversed the diet are predicted as if they are normal. So they have a metabolic profile in the hippocampus and in the cortex that resembles that of normal animals that have never seen high-fat diet or a high-sugar diet. This next graph is just the loadings of the metabolites in the PCA analysis for the hippocampus and cortex, and you can see that the most important metabolites are, for example, N-acetyl aspartate, which is a neuronal marker, taurine, glutamate also shows up here, glutathione, that's an antioxidant, and so on. There's some other interesting things, but what I wanted to call your attention to is that one of the metabolites that most strikingly changes with the high-fat diet or with the development of insulin resistance is the levels of taurine in the hippocampus. And we have seen that taurine accumulates in the hippocampus of these animals as they develop insulin resistance. And again, like the whole profile, it normalizes when we switch the diet. And we have seen this same effect in many other models of diabetes. We have pretty much all animal models where we measure taurine, actually all models where we measure taurine levels in the hippocampus, we have seen taurine accumulation in the hippocampus. And that's a beneficial effect. I'll not say more about it now, but there will be a slide on it at the end. So this was just to say that we modify metabolism in the brain, and if we normalize this diet, we recover the metabolic phenotype, and we also recover brain metabolic alterations in diabetes. And once more, that's not caused by glucose intolerance, because glucose intolerance happens already within 1 week of exposure to this diet, but we only see these metabolic alterations after insulin resistance is installed, in this case, at 4 weeks of exposure to the diet. What about memory impairment? So we did 2 memory tests at the end of this study, and we have this novel object recognition where we train the animals to recognize 2 objects. So in this case, novel location, and then we change the location of 1 object, and the animals tend to explore the novel location more. That's what we see here. So it's above 50%. But when they have that diet, they don't remember what's the new location or old location. And if we reverse the diet, that's normalized. And we see that both in males and females. And again, also with the novel object recognition, where instead of changing the location, we change one of the objects for a new one. So they prefer the new object. But in males, for example, you see here that it was not recovered, but in females, it did recover. So not everything recovers in diabetes, but we only tested after 2 months. Maybe if we would have tested longer, maybe that could have been recovered. We don't know. But there was certainly an improvement. And then we ask the question, so what are there, so we see that there are metabolic alterations. So are these memory impairment caused only by changing metabolism, or are we damaging brain structure as well? So we counted the number of neurons in the cortex and the hippocampus, so different areas of the hippocampus. And in fact, like in other animal models of diabetes that we have worked before, we have not seen changes in the number of neurons characterized by these new N positive cells in either area of the hippocampus or in the cortex. So neurons are not lost in diabetes, and we have seen that in other animal models as well. We have also looked at the number of newly formed neurons. So this double cortin staining stains for neurons that have that are generated in the and there are a few brain areas where neurons are made, like the subventricular zone or the dentate gyres of the hippocampus that we are staining here. And we could not see that the number of neurons decreases substantially, the number of neurons that are newly formed. So neurogenesis is probably not changed to a substantial extent in this animal model of diabetes. And then we want to look for glial cells as well. So the astrocytes and microglia. So we have stained for astrocytes. We have not seen changes in the number of astrocytes or in their coverage in the hippocampus and cortex. But what we have seen was changes in EBA1 positive cells, and those are microglia, the immune cells of the brain. You can see here that the number of cells of microglial cells has not changed significantly in the hippocampus and cortex. Neither the coverage in terms of total size of the cells so that's the top graph. So it's the area of EBA1 cells, and below it's the number of cells. So neither of those changed. But what we have seen is the number of cells that are activated, and that's what you have here in this graph on the right. So there is an increase in the number of activated microglia in the hippocampus and the cortex. When we normalize the diet, that is recovered. And activated microglia, they are usually characterized by overexpression of cytokines, because these microglia are increasing their activity to either protect the neurons, or they can be damaging the neurons as well. I'll talk about that in the next slide. And we have looked at the expression of cytokines in the brain, and we have actually not seen, this is in the hippocampus, we have not seen an increase in cytokines after, again, this is 6 months exposure to high-fat, high-sugar diet. I have to say that other studies have reported increased levels of cytokines early on in the exposure of high-fat diet. So within the first 2 weeks, there might be increase in the level of cytokines, but that is normalized afterwards, and it's not coming back at 6 months of exposure to these diets. Okay, so what about microglia? So microglia might have different activation states, and what I'm going to talk is about normal activation states of microglia. They can be resting, and when they are resting, they have this ramified morphology. They have fine processes, and they have an herborization of those processes, and they have a small cell soma. And when they have this morphology, what they are doing is surveilling what is happening in the brain. So they are just sensing, is there an infection that they have to take care of? They also produce some trophic factors. They are involved in synaptic pruning and keeping the homeostasis of synapses. But when there's an injury, when there's an insult, they can become activated. They retract the processes and they become more enlarged in their soma, and what they are doing is releasing cytokines and chemokines to attract other immune cells to come. And they are trying to recover homeostasis, so trying to protect the neurons somehow. They can also become phagocytic, where they become more abamoid, and what they are doing at that stage is they are proliferating a lot more, and they are involved in phagocytosis. So they are taking, for example, phagocyting some particles, for example, A beta, that could be accumulating and so on. And they have a pro-inflammatory profile. So what we are seeing in diabetes is desactivated microglia here in the middle. We have not seen phagocytic microglia in our animal models. But in contrast to what is normally seen in other diseases or other insults, we have not seen overexpression and release of cytokines. And we wanted to understand why, and to do that, we turned into a cell line of microglia. So what we wanted to do is to understand why obesity-associated microglia activation is not accompanied by overexpression of cytokines that is often characterized in this state. So Gabriela de Paula, that came to our lab some years ago, she came from Florianopolis, and she was characterizing BV2 microglia exposed to palmitate, which is a saturated fatty acid that exists in animal fat, and it actually accumulates in the brain of obese individuals as well. So what she has seen was that cells exposed to palmitate proliferate more. That's what you see here, the green versus vehicle. And we will also use a control experiment with LPS, that is an immunoactivator. So it's a bacterial stimulation, let's say. So we see this increased proliferation, and here you have MTT reduction showing that you have increased competent cells, so increased proliferation of the cells. But again, in vitro, we also don't see that palmitate increases the expression of cytokines. So the production of TNF-alpha, IL-6, IL-1 beta is not there, but it's seen in the exposure to a bacterial infection, so LPS. We have also measured the release of TNF-alpha from the cells. We also don't see that there's an increased release from palmitate. So also in vitro, these cells proliferate more. We know they change their metabolic profile. That's in this paper if you are interested. But they don't overexpress these cytokines. So how are they mediating? What are they doing? How are they mediating their information? So we thought about extracellular vesicles. So extracellular vesicles are particles released by cells as a communication strategy to other cells. And we thought that maybe saturated fat induces these activated microglia to release extracellular vesicles. And these vesicles will have a modified cargo that can act on neurons and other cells, such as astrocytes as well, and then transmit some kind of message. So indeed, so we collected vesicles from these cells in vitro and did a proteomic analysis and found that the cells exposed to palmitate had a substantially different proteome from those exposed to LPS or vehicle in vitro. And of course, we looked at what are those proteomic changes. And in a pathway analysis, what we found is that it's proteins mostly involved with processes that are associated with RNA processing and translation of proteins. So you can see listed here a number of pathways that are involved in the metabolism of RNA, of proteins, on translation, and so on. So it was all about those processes. Which is interesting because you can think that there is a proteomic or a proteome that is carried in these vesicles that is given to a neuron, for example, and will tell the neuron, okay, now you'll change your proteomic or your protein synthesis to produce these other kind of things, and then you'll change how you are functioning based on what this microglia is telling you. So now we still don't know, but are these messages good or bad? So are these messages an activation of microglia that is trying to protect the neurons, or is an activation that is damaging to neurons? So we took these vesicles from microglial cells and inject them in the ventricle of mice that were naive, so normal mice that were never exposed to any other thing but these vesicles. So they received vesicles from either vehicle-treated cells or from palmitate-treated cells. And guess what we saw? So we actually saw memory impairment. We did the same type of test. So this is a novel object recognition test. So during the familiarization phase, the animals have never met the object, so they don't distinguish between them. But when we give a novel object, they prefer explore the novel object, but not when they're exposed to the vesicles coming from palmitate exposed cells. So extracellular vesicles were able to convey a message that causes memory impairment in mice. These vesicles also caused, coming from palmitate, they also caused depressive-like behavior. So this is a sucrose splash test, when we measure how much time and how fast the animals start cleaning themselves after being splashed with the sucrose solution. And we can see that they are more delayed at starting to lick themselves, and they also take less time doing that. So they have some depressive-like behavior. And we did a glucose tolerance test, and I forgot to say it. So we analyzed this after 1 week of injecting the vesicles. And after that 1 week, we could also see the development of a little bit of glucose intolerance, which means that the vesicles injected in the ventricle might also reach the hypothalamus and mediate something there, and then cause some metabolic impairment at systemic level. Okay, so this is a summary slide of what I've been saying. So in diabetes, or let's say in the brain, we have these glial cells, astrocytes, and microglia that are involved in supporting neurons and protecting neurons. But in insulin resistance and in type 2 diabetes and obesity-induced type 2 diabetes, we have a loss of these supports. So there's a loss of function. And at the same time, we have some toxicity coming, especially from microglia on the neurons. So we cause neuronal dysfunction. And what we want to do is to find ways of reversing these. So can we do that? And I'll just have a slide that summarizes a number of experiments that were done to do that. We are working mostly with dietary interventions, and we have published a paper a few years ago on high-fat diet-exposed animals, and you can see that there's a memory impairment. But when we treat them with taurine, you remember that taurine accumulates in the brain of diabetes, and we think that's protective, counteracting protective action. So by giving taurine in the diet, we can prevent memory impairment in the animals. When we give N-acetylcysteine, which is a cysteine donor that boosts antioxidant buildup, especially in the form of glutathione, we can also recover from memory impairment induced by high-fat diet. We have done experiments with caffeine. Caffeine is an antagonist of adenosine receptors, and these were studies in the past where we had seen that adenosine receptors are modified in diabetes, so we can target them to protect, and we did that with caffeine. And you can see here that in the model of insulin resistance, the Goto-Kakizaki rat, we have memory impairment in a Y-maze spontaneous alternation. And when we give caffeine, we can improve that memory performance in these insulin-resistant animals. And we don't work with exercise, but I thought that in the context of this symposium, it would be interesting to tell you that also as exercise exposure, which is it's probably one of the best insulin sensitizing interventions that one can do, can also recover to some extent memory impairment, in this case in obesity-associated memory impairment in rats. This is not work from our lab, but it's also an interesting study. My last slide is just to tell you some key messages that impaired memory in type 2 diabetes is likely the consequence of metabolic and functional changes, but not structural alterations that lead to neuronal loss, because we don't see neurons dying in our animal models. We see that diabetes and metabolic syndrome factors impact other cells than neurons, and that's mainly astrocytes and microglia that we have worked with. And there are brain alterations in diabetes that can be prevented by improved lifestyle interventions in animal models. And we have been working with dietary interventions, but there are also exercise paradigms that can improve brain function in animal models. And finally, what we need to do urgently is to translate our findings to human populations. And at the moment, we started working with some cohorts that exist here in Sweden to try to understand which are the diabetic individuals that would mostly need interventions, and how can we translate our interventions into humans and help diabetes patients. Okay, this is our group and our collaborators, and thank you for your attention, and I'm happy to answer any question. I hope I'm within time and didn't take too much.
Obrigada, professor João. Deu tudo certo com o tempo, sim. A gente vai ter o espaço para as perguntas agora. Obrigada pela sua apresentação. Realmente é muito interessante o seu trabalho. O pessoal gostou bastante. E eu vou passar aqui para algumas perguntas, tá? A primeira pergunta do Davi é: qual é o impacto das flutuações de glicose nos níveis de concentração e na memória de curto prazo?
A concentração de glucose no cérebro, é essa a pergunta?
Isso,
É,
na memória de curto prazo.
hum, uh, eu tenho a sensação, eu não sei, tenho a sensação que alguém fez essas experiências e eu lembro-me de ver um artigo, mas não me lembro. O que eu vou dizer é o seguinte: a glucose no cérebro depende eu vou dizer isto em inglês, é mais fácil. Glucose in the brain is function of plasma glucose. So as you increase glycemia, you are increasing brain glucose, and that's the relation of 1 to 4. So there's 1 millimolar of glucose to 4 millimolar of glucose in the plasma. And I don't think that of course, if you don't control glycemia, you'll have high glucose levels in the brain. I don't think that the courses of glucose during a glucose tolerance test, so after a meal, if they are so these short glucose transients, they are not a problem for the brain. The problem is when this glucose persists high for a long time in the individuals, and of course there you have deleterious effects on the cells. Just because glucose itself is toxic if it persists high. That's actually, I'll just say something that is important, is that most studies in cells in vitro use high concentration of glucose in the cell culture medium. Often the neurons are kept in 25 millimolar of glucose, and that's not necessarily a normal neuron in the brain. So some care needs to be taken when looking at those kinds of experiments. I'm not sure if I answered the question.